So cool. I guess this is the second to last day. And so by this point in time, you guys have gone through and done some basic BCL stuff, filtering molecules, processing molecules, aligning them, computing descriptors, hopefully generating data sets. And I'm sure a lot of you have also trained some neural networks and done some QSAR and maybe even some QSPR. And then yesterday, we started to get into the reaction-based drug design and a little bit of the introduction to the alchemical framework. And so reaction-based drug design, once again, using the reaction framework in the BCL, which was originally designed for ligand-based drug discovery by Alex Genes, but which we've been modifying to make it extensible to multi-component reactions in both a ligand and a structure-based context. And then also the alchemical framework, which is really less of, it doesn't give you a route towards synthesis, but what it does is let you sort of explore the chemical space in a lot of different creative ways with some interfaces that are meant to try to mimic some medicinal chemistry type intuition. And so it doesn't matter if you've done both of those or neither of those or anything like that. We're going to keep sort of trucking ahead, and as you get to the tutorials, just feel free to ask questions and whatnot. So today we're going to mix a couple of things together. So the first tutorial is going to be on chemogenetics, which we'll describe in a second, and the second tutorial is going to be on selectivity design. Both of these are just tasks that allow us to not just sample the chemical space of the small molecule, but also sample some of the sequence space of the protein we're working on in different contexts. And so the idea is that there really aren't any other software packages outside of Rosetta that allow you to rapidly sample sequence space in the context of a backbone, let alone do it in a bunch of different customizable protocols. And there are a few different contexts in which you might want to do that with a ligand present, right? Especially if you're designing a protein around a small molecule, or you're doing enzyme design, or you're doing anything like that. It's important to be able to handle having a small molecule there, but we're going to sort of extend that to say, how do you actually change the small molecule while you're changing the protein? And like I said, there's a few different times in which you'd want to do that. The first tutorial is going to really focus, the first tutorial being tutorial number 6, just on sort of reinforcing some of the ideas from tutorial 5 with respect to the alchemical framework. It's also going to be teaching some tips and tricks related to stereochemistry and some other things. And so just taking a slightly bigger step in sort of learning how to handle that framework. Tutorial 7 is going to also teach you new tips and tricks with respect to the alchemical framework, but more with respect to the ring swap mutate and all the different ways you can work with it. And so part of these tutorials is sort of trying to get you more familiar with the mutates, and then the other part of it will be, in the case of tutorial 7, we're also going to finally do our add medchem hack for the one-shot, made on demand type synthesis. But before we get to that, let's go ahead and do tutorial 6. And so, like I said, tutorial 6 is about chemogenetics. And I mostly want to share my screen and talk about this. And so we're going to start up here. This is sort of derived, there's a publication in 2018, I think it's by Runchi et al. I hope I'm pronouncing that right, but if not, it's a really great paper. And what they do is they show optimization essentially of a small molecule and a mutant protein pair to increase the selectivity of the interaction. And so oftentimes we have proteins in some system that we don't know the function of, or that we want to investigate independent of homologous proteins, and it may be challenging to do so, because oftentimes we cannot just knock out these genes in animals and see what they do phenotypically, because maybe there's some side effect with, in this case, gene transcription that makes that challenging. Well, the other option is to maybe pharmacologically knock out the protein of interest, but as many of you may know, it's challenging to develop selective inhibitors, especially when you're trying to design a selective inhibitor for a particular homologue in a whole family of proteins. And so the idea with chemogenetics is, why don't we simultaneously perturb the small molecule and the protein of interest so that we can create sort of an optimized pair of interactions between the two of them. And so the central sort of hypothesis is that the perturbation you make to the protein does not change the protein's function. And so usually you have to do some type of control to make sure that when you perturb the protein, you are not you're not destroying the function you're actually trying to investigate. And then at the same time, you want to make sure then that your small molecule, you're changing it in a way that is still going to be selective for that new mutant. And there's other applications for this. So in the neuroscience community, there's these things called DREADs. And so there are receptors that you design to be specifically activated by a particular small organic molecule. And these can be really useful when you're trying to express proteins, usually to control sort of the activity of neurons in different systems. And so this is sort of an interesting field. It's a very, it's sort of a research probe development field, but it's really cool. And so the idea is, can we simulate these things in Rosetta? And so the tutorial today that you guys are going to be working on is going to be this. This is BRD2. So this is a bromo domain protein. It's a transcription factor regulator, I guess. I don't know. I don't personally, honestly know all that much about this protein. And there's already been some really nice work, and I've got some citations in the tutorial. So you've got the links to the primary literature for the development of this sort of scaffold right here. And what some of this work has done is investigated a couple different mutations at this position right here. So this is leucine 383 in the sort of native wild type numbering, or leucine 41 and the numbering we're going to be using. And then this is a scaffold for the molecule. And the strategy that they take to do these chemogenetic studies is called the bump and hole approach to design. And so sort of the plan then is to say, okay, well, if we want to make an inhibitor selective for a particular protein, what we can do is we can take the protein, and we're just going to do some sad little pymol mutations for the moment. You can take this residue, this leucine, and maybe you bump it down to an alanine. Well, what you've done is you've created a hole. You've created more of an opening by removing a hydrophobic. And so then what you want to do is grow your small molecule into sort of the cavity that was created by the change of a larger amino acid to a smaller amino acid. And so you're bumping your small molecule into the hole created from your substitution. So it's the bump and hole approach for chemogenetics. And so that's great. We can do that, experimentally. But it'd be nice if we could also simulate that, so that way, like everything else, we can test fewer pairs in the wet lab. And so today in tutorial 6, if you go to the scripts directory, we've got a bunch of scripts here. We're going to sort of walk through setting this up for the BCL Rosetta integration. And so today you're not actually really going to be using BCL standalone all that much. It's going to be pretty much entirely using Rosetta. And so the idea is we can go down. All these movers are probably familiar to a lot of you. We've got, standard, scores. We're in this case using ligand. You could also use Talaris 2014 if you want. Residue selectors for the ligand, the receptor, the interface between the two. We've got some task operations to restrict certain interfaces. And then we've got our nice filters for the interaction energies, all normal stuff. If you made it to tutorial 5 yesterday, you may also start to be familiar with the BCL drug design movers. And so we talked about BCL fragment mutate mover. And in this particular example, it's actually really easy because the fragments that we're going to be using are just fragments that we can derive from that manuscript by Runchi et al. That did the optimization of this bump and hole approach for this particular protein. And so what I've done is I've already sort of created several directories of fragments for you folks. And so you can see we've got our bump medchem fragments for position 1, got our bump medchem fragments for position 2, and then we've got a methoxy fragment that we're going to use to scan sort of part of the molecule. And so this is very simple. We only need to use one mutate. We're going to be using the add medchem mutate. My XML, it's all funky looking. Anyway, and there are a few things then that we can focus on since we're doing this. The first thing we can focus on, if you hadn't thought about it yesterday, one of the fun things is we can define as many AdMedChem movers as we want. We do not need to just limit ourselves to one. And you can make each and every one of these things target a different subset of atoms. And if each of these targets a different subset of atoms, then what you can do is have each of these subsets of atoms receive different medchem library fragments. And so the AdMedChem mover that's going to act on mutable atoms 16 and 17 is going to be passed a variable that corresponds to that methoxy fragment. And so this is going to allow us to only scan that particular fragment on these two positions. The other thing to note here is that these two AdMedChem mutates actually both correspond to the same mutable atom index. If we go back to that IML session that I had, let's see, it's up here somewhere. Yeah, not this one. That's okay. This is the one. One of the things you'll notice is that, and I showed it a little bit differently in the tutorial just because I was trying to be consistent with the manuscript a little bit more, but the important thing to know is that we are adding our methoxy fragments to one of these two residues right here. So we can color these. So that's where our methoxy placement will be. And that's fine. That's just an aromatic ring, no big deal. That'll definitely correspond to what we think. But this one right here, this is our bump segment. This is where we're going to be adding, oh, that looks like an oxygen. That's bad. Shouldn't do that. Slate. And so this is where we're going to be adding our functional groups for the bumps. And we're actually adding 2 bumps here. And so then the question is, how do you control the stereochemistry of the resulting compound? And I think we talked about this a little bit the other day, but this tutorial is going to go through it in more detail. We have a few options. First, what I want to point out is that by default, recall that we have to remove as we go. Let me set label size. We have to remove these hydrogen atoms in order to append additional fragments to this atom, right? Otherwise, we create a non-gas Eiger atom type. And so the default behavior in the BCL is to proceed in order of the lowest hydrogen atom index. So we go to this atom 17 or whatever, which is going to be our carbon, and we're going to start picking off atoms, hydrogen atoms, and appending to that position. Well, we don't always want to go in that particular order. Let me rephrase that. I should say by default it randomly chooses a hydrogen atom, but the default, if you are not randomizing, is to start from the lowest and go to the highest. And so we can control just sort of the sequence in which we run through these. You know that you can, though I don't usually recommend it, specify hydrogen atom indices directly. But what we can also do is specify this thing right here. So this flag is the open valence shuffle H flag. And this is just part of our util object data label initialization in our mover. And so Rosetta will interpret this the way the BCL interprets it, because Rosetta is just making one of these BCL objects and applying it to a molecule that will then be converted back to part of our pose. And so this open valence OV shuffle H is set to false. That means we're not going to shuffle. We're not going to do a randomization. We're going to proceed from lowest to highest. You can, however, say OV reverse. OV reverse, when you are not shuffling, is going to say, instead of proceeding from lowest to highest, proceed to from highest to lowest. And so that gives you a little bit of directionality for where you're going to be adding the substituents on your molecule. And you'll play around with that a little bit during the tutorial. Okay, so these are some some examples that we're going to run with AdMedChem. At some point, what I'm going to have you do after you go ahead and try to recreate the benchmark from that manuscript I've been discussing, we're going to take it a step further. We're just going to say, hey, can we use Rosetta to redesign the binding pocket to bind to, well, I should say, to redesign the binding pocket while also redesigning a small molecule using the, the fragments from that manuscript. And if you really want, you can also use more fragments. I don't really care all that much. I thought what might be fun is to say, you know, we'll take maybe the best designs and we can run some MD simulations of them and have a little internal competition to see who's got, you know, the best MMP-BSA binding affinity or something. So if that sounds appealing to you, I'd be down to do that. I think it'd be fun. But you know, you can write your own protocol to do it. You can use the protocol that I showed you right here, where we've got a little tiny generic Monte Carlo mover that is going to repack the protein after you design a small molecule. So for every design of the small molecule, you'll perturb the protein around it, both in sequence and sort of position space. And hopefully after you do this and make a bunch of small molecules and a bunch of proteins, we'll have some good ones. I did a few fun designs. I have a couple pictures of them, of two of them in the manuscript, or not manuscripts. There's no manuscript yet. In the tutorial. And so, you know, we can go ahead and we can look at them and say like, okay, well, here's one of the designs. How is this different from what, how is the sequence as well as the inhibitor different from what we saw in the run sheet paper? Because you're going to sample more sequences, and so you might find that some of the modifications they had proposed work better on sequences that they don't actually have. I've noticed that Rosetta likes to take some of these residues over here and convert them into polar residues and can form hydrogen bonds with a few different substituents in the ring right here. So anyway, I think it's fun. You don't have to do it, but I think it's fun. And that's sort of the idea. So we're going to try to design the protein at the same time as designing the small molecule. I've got a few minutes. I think we ended a little bit early because this is a pretty fast talk. I'm happy to take questions before we get started, or I can let you guys be free and I'll just stick around to help as people run into problems. Again, the second tutorial today is going to be doing sequence design, but in the context of computing selectivity while you're designing a small molecule, and then using using the small molecule perturbations to try to optimize the small molecule for selectivity. And then after you do that a few times with the standard alchemical mutates, we'll do it with an amide coupling reaction using some sample reagents that you might find from a made-on-demand library. So that's all I got.
Hey, Ben, I have a question about kind of perhaps the application of this in real life chemogenetics. So if I have, so you pick a specific residue in the, or a specific atom in the ligand to grow, is there a way to randomize that in a Monte Carlo fashion, perhaps, so that you're just growing depending on, I guess I'm thinking about having, you know, not just designing a protein, but taking a protein that has, you know, a couple hundred patient mutations and trying to design a drug that could bind to all of the patient mutations, or as many as possible.
Yeah, so that's a really good question. I will say that the metric that you want for your example, so if you've got 100 proteins and you want to make a drug that binds to all of them, there's a metric called the selectivity entropy, which is a measure of how much does my drug bind to a bunch of different proteins. And we often use it as a selectivity metric for, you know, how promiscuous is our inhibitor for all these different kinases or something, right? Are we binding all the kinases or are we selective? Usually we'll want to be very selective, but in your case, you want to be very pan all these mutants. And so we can compute that in Rosetta scripts if you want to, or in PyRosetta. And yeah, we can do that during design. With respect to randomizing the small molecule, yeah, we can. In this example, the reason why we're doing these sort of sequential growing schemes is really just because we're trying to replicate the benchmark from this particular paper. Not benchmark, really. Replicate the experiments from this paper. And during medicinal chemistry, like oftentimes you just want to run through a molecule and do scans on different parts with, you know, halogens, methoxys, you know, esters, things like that to see what fits and what doesn't. And so there are different cases in which you might want to do more randomization, more large perturbations. There's all kinds of different optimization algorithms outside of MCM that you can use as well. So, but yeah, if you want to design an algorithm that's going to sample different parts of a molecule for a bunch of different protein mutants, we can definitely do that.
Thank you.
Ben, another question? Maybe?
Yeah.
So what about the sort of covalent bump and hold?
What about the covalent one?
So can it be simulated in the using the BCL Rosetta? Like I'm guessing no, probably. Like for example, mutating something like a residue to cysteine and adding like a covalent warhead as the bump, sort of, because like there are examples like this in the literature, but it's not probably as known as the normal bump and hold.
Yeah, that's an interesting idea. I will say that for the
just to correct myself, I'm not sure there are examples like this in the literature. But
that's okay. But the point about covalent inhibitors is well taken, right? And so in tutorial 4, we go through how to do the sort of reaction-based design where we try to keep an acrylamide warhead in an appropriate geometry to form a covalent inhibitor. And so, so typically you predefine where you want the covalent warhead to be, and you build around that. I see. But we could probably do something more sophisticated too, because I know you've done a lot of stuff with this. Like, if you design a drug, then is that part of the drug able to form a covalent bond? I'm sure there's a way to work that in algorithmically. So yeah.
Okay, but yeah, this tutorial sounds fun. So I'm looking.
Cool, man. Appreciate it. All right. So if anybody wants to do the competition to see who gets the very best code designs and run MD simulations, let me know, and we'll have like a friendly little thing. But if not, it's also fine. We don't need to use GPUs. Cool. All right. Well, thanks everybody for your attention. I really appreciate it. Have fun. Let me know if you have questions as you go. Yeah. Talk to you later.